This is part 3 in our series on transcription and translation, and today we'll be focusing specifically on part 1 of translation. There are 3 distinct stages of translation. There's initiation, where the ribosome binds the mRNA at the start codon. There's elongation, where the nascent polypeptide elongates by the addition of amino acids. And there's termination, when the stop codon is translated and the peptide releases from the ribosome. In part 1 here, we're going to focus on kind of getting oriented and set up with the various players and concepts involved in this big process of translation. In the genetic code, each of the 4 nucleotides A, U, C, and G are combined in various ways, 64 to be exact, and they will create 3-letter sequences called codons. However, only 20 amino acids actually exist, meaning that several codons will specify a single amino acid. These codons specify various amino acids and/or start and stop codons. Notice here that AUG, which codes for methionine, is a universal start codon in eukaryotes, while UAA, UAG, and UGA are stop codons. Each amino acid can be grouped into polar and nonpolar, charged and not charged categories. Each of these triplicate nucleotide sequences are referred to as codons or as open reading frames. 3 nucleotides don't automatically find and bind their amino acid. This process is mediated by adapters called T or transfer RNAs, and these RNAs fold into very specific shapes that kind of look like clover leaves. And they bind the codon here with an anticodon loop, and they will recruit and snag the proper amino acid to bind onto the nascent polypeptide. The genetic code is redundant, like we've discussed, meaning several open reading frames can code for the same amino acid. So logically, there is 1 amino acid for every nucleotide set that codes for an amino acid, regardless of the variations. Some amino acids have their own tRNA, but some have a tRNA that includes a wobble position. That can be shown here, and here, the 3rd position. This is when the last nucleotide in a codon can be mismatched or wobbled. Here, it's shown by a leucine tRNA, and we see G is pairing with C, A is pairing with U, and then G is pairing with C, which is normal Watson-Crick base pairing. But then you also see in the wobble position, G can bind with U. So in these wobble positions, this is how we see so much variability in the genetic code. So how does the tRNA get paired up with an amino acid and primed for transcription? Amino acyl tRNA synthetases covalently couple specific amino acids to their tRNAs. In this example, we see that the synthetase here in blue binds methionine, ATP, and the tRNA for methionine. This process includes a 2-step hydrolysis, which includes ATP losing both phosphates. The synthetase uses hydrolysis and ATP to convert the amino acid into an adenylated amino acid, hydrolyzed again, and now the amino acid is aminolated and bound to the tRNA as well as charged. This is now our product, charged tRNA, methionine. The correct amino acid has the highest affinity for the binding pocket, as we saw in the previous slide. But if the wrong amino acid binds into the catalytic site, there are several different editing mechanisms later on that will ensure the correct tRNA is bound to the correct amino acid. The synthetase will do a double check after AMP is bound and try to shove the tRNA and amino acid into another editing pocket in the enzyme. And this process is called hydrolytic editing. The AMP is removed, and if it doesn't fit, the process will start again. Here, threonine did not fit, but serine did, and now the hydrolysis is complete. The whole process of translation is facilitated by ribosomes, which are just massive complexes of about 50 plus proteins each. Here's the mammalian ribosome. It's very, very large, and it's made up of the large 60S subunit and the small 40S subunit, each of which have many different rRNAs and proteins. To coordinate this huge event of translation, the ribosome has highly specific structure that contains 4 different binding sites. One of these is for the mRNA, and then 3 of them are for tRNAs. So there is a E site, a P site, and an A site. So how does the ribosome know where to bind onto the mRNA and start translating? It will bind the 5' cap of the mRNA and slide along until the RNA reaches the start codon. AUG is the signal to kind of orient and begin peptide synthesis.